Good morning, everyone. My name is Luke Townsend, and today we're presenting my master's thesis work with Monkeyface Pricklebacks. So freshwater eels are popular in many seafood dishes, like sushi, within unagi, but this popularity has led to declining in their population significantly as they're overfished to meet that demand, resulting in some species actually being classified as critically endangered. As can be seen over here in Europe, it's that shaded red region. The European eel is classified as critically endangered. Recovery of these species has been poor because the overfishing of those juvenile freshwater eels has substantially decreased their natural recruitment. Freshwater eels are also catadromous species where they have a complex life cycle and their larval recruitment is dependent on multiple environmental factors, making recruitment pretty difficult for them. So the combination of overfishing and that reduced natural recruitment has led to a low supply of freshwater eels out in the wild. So one option is, can we culture freshwater eels to supplement their eel supply? Currently, we're having difficulty reproducing the eels in a captive setting, so right now that's not really a viable option. Research is being conducted, but right now the supply for freshwater eels is based on wild catch. Additionally, freshwater eels are carnivorous finfish species where they have to consume diets containing fish meal and fish oil, which are byproducts of reduced forage finfish, and it's not really sustainable to feed one fish from another fish population. So another option is to culture an alternative fish species that has a similar morphology to the eel, so you can supplement it to that eel fishery and something hopefully a bit more sustainable with a different diet, so not carnivory. And Monkeyface Pricklebacks might have that potential as an alternative species to freshwater eels, as they're an eel-shaped intertidal cicadidae. Largest of the prickleback species, around 0.8 meters in length. They have a wide range from Baja, California, all the way up to Oregon, and they undergo an ontogenetic shift, shifting their diet from carnivory to herbivory, around 7.9 centimeters in length, where they increase their gut length, and the alpha amylase activity increases, so they're able to digest the starch content of macroalgae. And so that's what I want to highlight is they're capable of consuming alternative and sustainable ingredients. They also have several appealing characteristics warranting an investigation as an alternative species for aquaculture. The first being which herbivorous lifestyle, they can consume a more sustainable diet. The next being which they have a potential high stocking density, so multiple individuals can be found within the same crevice out in the intertidal zone. Third, they lack a large-scale fishery, so they might be more socially accepted as opposed to other finfish. They are adapted to the intertidal region, so they're tolerant of fluctuating environmental conditions like temperature. It's actually been found that the species can remain outside of water for 18 to 37 hours if kept wet. Static respirometry work has been conducted with the species as well, and it's found that they have a low metabolic rate in comparison to bald sculpin, which is relatively similar in size. And based off that low metabolic rate, it suggested that Monkeyface Pricklebacks are efficient in their energy usage, and so they have a potential low feed conversion ratio, so that conversion of your feed to that biomass. It's also been found that they have a potentially rapid growth rate. After undergoing that ontogenetic shift, they grow at about 5 millimeters per month. And then lastly, a past student at Moss Landing Marine Laboratories conducted some reproductive trials with Monkeyface Pricklebacks, and he was able to rear them in a captive setting, and he had a successful clutch with a 1-to-1 ratio of males to females. However, we have some knowledge gaps, specifically the optimal diet and temperature to rear these species to achieve a maximum growth. And so those are the objectives of my master's thesis, so identifying the optimal diet and temperature. And I examined growth, feed efficiency, condition, energy reserves, fillet content, so macronutrients and the fatty acids, metabolic rate, standard metabolic rate, metabolic cost of digestion, your specific dynamic action, and then thermal limits, so your critical thermal maximum. So the first time we were talking about the feed study, and so we went down to San Simeon, California, and captured multiple juvenile freshwater eels, brought them back to Moss Landing Marine Laboratories, acclimated them onto our flow-through system, and we had 4 feed treatments. So you had your leafy algae as their natural diet. Moving on to your algae-based pellets. So pellets are popular in the aquaculture industry because they have many different benefits, such as improved feed conversion ratio. So looking at the pellets as well. And then we moved on to a mixed pellet. And so it's a mix of algae and soy protein concentrate. Because Monkeyface Pricklebacks are herbivores, they have possibly adapted to consume terrestrial-based ingredients. And looking more to soy-based pellets, so mostly composed of soy with a little northward palatability. And so the previous master's student, Matt Hohen, found that these species can be a little competitive. And so the larger individuals were outcompeting the smaller ones. So to remove that potential artifact, we have, within each treatment, they're in triplicate sets, but this tank is only small-sized fish, medium-sized fish, large-sized fish for each treatment. And so this study was conducted over 11 months, and for the first 4 months, we collected biweekly growth measurements and daily feed intake, and the remaining 7 months was monthly growth measurements. And throughout this, we measured metabolic cost of digestion, that specific dynamic action, also looked at proximate analysis. And so here's a breakdown of those feed treatments. I'm not going to go down the exact formulation of the pellets. I have that later on if anyone's interested. This is just the breakdown. So you have your leafy algae, your ova species, Devilarius Pacific dulce, and then you have your nori, no soy protein concentrate, and then your algae-based pellet composed of those 3 different types of algae. And then your mixed pellet, same thing, but it's including some soy protein concentrate. And then the soy pellet composed mostly of soy protein concentrate, but a little bit of nori for palatability. And so now moving on to the results of this feed study. So based on a generalized linear mix model, we got diet on the x-axis, and you got specific growth rate. On the y-axis, you can see that the pelleted treatments, those fish had higher growth. And the soy pellets, actually, those fish consuming those soy pellets had a higher growth than the algae pellets. And because it was done and conducted in a flow-through system, I also looked at thermal growth coefficients to kind of account for that temperature influence. And I found a similar result without the temperature influence. It's the same pattern and similar growth rates. And it should be noted that the highest SGR was around 0.2. I'll bring that back up later. So looking at condition indices, so Fulton's K, based on a generalized linear mix model, fish consuming the pellets were in better condition than those consuming the leafy algae. For feed conversion ratio, the lower FCR you have, the more efficient you are in converting that feed to biomass, and found that the fish consuming the pellet diets were more efficient than those consuming the leafy algae. And then looking at metabolic rates, so you got your standard metabolic rate up here, and your specific dynamic action down there with one-way ANOVAs. Found that pellets have similar metabolic costs with the leafy algae. And so those growth differences that we just previously observed were likely not impacted by those metabolic differences. And so looking at the conclusions of that feed study, the mix in soy pellets, in terms of like a growth perspective, were optimal diets. We also found, yeah, they like swimming around a lot. But in comparison to those European eels, those growth rates were a bit slower, as 0.2%, as opposed to 0.6%. However, that study with the European eels, those fish were reared at around 25 degrees Celsius. The average temperature for the Monkeyface Pricklebacks in our study was around 12.5 degrees Celsius. So temperature might be the key in order to grow the species a bit faster. And that was my next study. So similar collection methods, going down to San Simeon, bringing them back to Moss Landing, acclimating them to that flow-through system. And then we fed them the soy-based pellet because we're trying to maximize growth, so that from the previous study. And reared them on 5 different temperature treatments, so from 14 degrees Celsius all the way up to 26 degrees Celsius, with 3 degrees Celsius apart for each treatment. And we did that with this setup right here because we had a flow-through system. So we used a heated water jacket, so water would flow into this trough, it would heat up, and then it would be pumped into each tank, and then they'd flow out through the one trough, so they're not sharing water. And that's how we were able to achieve our temperature treatments. And so biweekly growth measurements were conducted and daily feed intake over a 3-month period, looking at metabolic rates, critical thermal maximum, and proximate analysis work as well. So first looking at the standard metabolic rate, based off a nonlinear regression, you have temperature on your x-axis and standard metabolic rate on your y-axis. You can see an exponential increase in that standard metabolic rate. And then if you look at that feed intake, where you got total tank weight consumed per day, you can see that as temperature increased, they increased their feed intake, and that was to kind of satisfy those metabolic increases. So in order to grow, you need to have a caloric surplus, so they increased their caloric intake. And then now if we're looking at the growth rate, based on the generalized linear mix model, you can see that we had an optimal growing temperature at 20 degrees Celsius with an SGR of around 1. But when you got to warmer of a temperature, metabolic rates kind of exceeded, came too high, and their growth rate decreased. And so you get the highest growth at 20 degrees Celsius and lowest growth at 26. And looking at their Fulton's K, you can see, based on a generalized linear mix model, that 20 and 23 degrees Celsius were best for condition. There's no significant difference between these two, but fish reared in the highest and lowest temperature treatments had the poorest condition. In looking at the critical thermal maximum and how it kind of shifted with an acclimation temperature, you can see, based on a nonlinear regression, that fish reared at 14 degrees Celsius had a critical thermal maximum around 30, and as you increase the acclimation temperature, it kind of peaked at around the 23, 20 degrees Celsius with a CTM of around 32.5. But once you get to 26 degrees Celsius, the CTM actually decreased, and this was likely due to the fish being in poor condition at the time. This was conducted roughly about 2.5 months into the 3-month study, and so fish had already lost a lot of weight at that point. And so yeah, the conclusions of that temperature study are the thermal limit for this species is capable of shifting. So monkeyface pricklebacks are capable of growing in a warmer climate, unlike stenotherms. And then we found an optimal temperature to rear the species at 20 degrees Celsius, and accounts for those increased costs. So like as you're increasing your temperature, they're going to consume more feed, so you have to feed them a lot more. However, so if you're rearing them at 12 degrees Celsius, you're going to feed them for 1,500 days. But if you rear them at 20 degrees Celsius, you're only rearing them for 286 days, so accounting for that feed cost. And then in comparison to the European eels, we can see that we had a higher SGR at 20 degrees Celsius. So just overall conclusions, looking at the results of both of those studies, monkeyface pricklebacks are a potentially viable and sustainable alternative to freshwater eels, as they're capable of growing 383% in a year at 20 degrees Celsius on a soy-based pellet. But future work needs to be done, such as reducing that competition that I noted earlier, reducing diseases and parasites that we found throughout the study, extending that growth study with the temperature, trying to see like at 20 degrees Celsius, are they capable of maintaining that growth rate for an extended period of time on the soy-based diet and also the algae-based diet. And then kind of closing the life cycle of the species. And so I'd like to acknowledge these different funding sources: San Jose State University, NOAA, Sea Grant California, Moss Landing Laboratories, USDA, UC Davis. And then fish husbandry takes up it's a lot of work and help is needed, so I'd like to acknowledge all these different people for this would not be possible without them. And so now I'll be taking any questions. Yeah, they do taste the same, yeah. Yeah, I couldn't really eat the fish in this study, but we did have some adults, and we were able to taste them, and they do taste pretty well. And the one part of the grant was actually taking the adults, bringing them to like some chefs, and trying like a blind taste test. And so I think they're still working on that right now, but yeah. Mm-hmm.
Really cool talk. I'm excited to try this one today.
Yeah.
But I'm curious about your feed study.
Mm-hmm. Yeah. So. Yeah. So I can actually pull up. Yeah. Here's the pellet formulation for that. So based off a calculated analysis. So he was asking for like the kind of like the formulated breakdown of the diet study. And so yeah, we did it based off of isolipidic and isonitrogenous. Looking at the crude protein, you can see it's around 31.6% between those 3 different pelleted diets. And then what we're really changing in that would be that soy protein concentrate. In the kind of algae content of that. Does that kind of answer your question? Yeah. Okay, yeah. Thank you.
So I might have missed this, but in terms of spawning them, where are you guys at with that and actually breeding them in captivity?
Yeah, so he was asking a question about in terms of spawning monkeyface pricklebacks in captivity. And so Matt Hohen was the previous student conducting a lot of those reproductive trials, and he was able to find a 1-to-1 ratio with males to females to get a successful clutch. And then he reared a couple to larval stage, but we didn't have quite the right setup, so they didn't get them to survive that long. And so we did keep them, the adults, around a bit longer that had that successful clutch. And they were able to have another clutch. And so instead of this time taking that clutch out of their habitat, we let the parents take care of it, kind of aerating it. Unfortunately, about I think halfway through, the male got into it and just tore it all apart. So not quite sure what was going on there. I mean, it was being fed, so there's going to be more research conducted there. But yeah, great question. Yeah. No, they are not true eels. So they're of the prickleback species, and they do not have leptocephalus larvae.
And that's the big challenge to the European American.
Yeah, just more research needs to be conducted in kind of like reproducing the species. But yeah, they don't have that leptocephalus where they're impacted by like the different environmental conditions and stuff like that. But yeah.
If you have any other questions, you can address them later. We have to proceed. Thank you very much.
Thank you.